As we go through this group of courses, we're going to focus on the science behind each and every one of the steps of the Western blot protocol. But for now, on this first course, we're just going to do a brief overview of those steps, and we're going to start by telling you that the Western blot protocol can be divided in 3 main parts. On the first part, we're going to have to extract proteins from any given biological sample. Now, usually proteins are isolated from cell lysates or tissue lysates, but really they can be extracted from any biological sample. Viruses, bacteria, plant, anything that's biological. Now, once we have extracted those proteins, we're going to have to isolate them from everything else that was on those cells. For example, RNA, DNA, lipids, they are all going to be considered contaminants on our Western blot protocol. So we really need to make sure we can isolate our proteins so that we have a successful protocol. Once we have accomplished that, it's time to estimate the concentration of those proteins so that we know exactly how much protein we're going to load on the gel. At that point, we're going to load our samples on the gel and run an SDS page. SDS page stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis, which means I'm going to load those proteins and then I'm going to use an electric current so that they can run through the gel and be separated by their molecular weight. Part 2 of the protocol encompasses transferring the proteins from the gel onto the membrane. And that step is actually pretty important because it's going to facilitate the primary antibody in recognizing the antigen. It would be very hard for that primary antibody to find its antigen in a gel matrix. It's going to be much easier when those proteins are on a membrane. So to accomplish that, we're going to use once again electric current to help the proteins to move from the gel onto the membrane. And those proteins are actually going to bind not only to the surface of the membrane, but also to its inner structure. Once the proteins are bound to the membrane, we're going to take advantage of primary antibodies that are developed specifically against certain antigens so that we can detect our target of interest. Now, if we think about it, there's thousands of primary antibodies, and it would probably take a long time and a lot of resources to label thousands of antibodies so that we can directly see our target of interest. So scientists thought about that puzzle there and decided that perhaps we could use a secondary antibody that recognizes the primary antibodies and simply label those secondary antibodies. That way, we would only have to label a handful of antibodies, such as our anti-rabbit, anti-mouse antibodies, and so on. Now, when Western blots were first developed, the secondary antibodies were labeled with enzymes, such as horseradish peroxidase, also known as HRP, or alkaline phosphatases, known as APs. Now, depending on which substrate we used with those enzymes, we could either end up with a colorimetric Western blot or a chemiluminescent Western blot. That would depend on the enzyme and the substrate that we used. Now, on the colorimetric Western blot, we actually see the color develop on the membrane so we can see the bands with our naked eyes. On the chemiluminescent Western blot, photons of light are produced, and we can then capture those photons of light either with the aid of an X-ray film or with a specific imager that can see and translate those photons of light into a digital image. Now, it can be a little bit challenging to depend on an enzymatic reaction because that reaction changes with time, with temperature, and a lot of different factors. So as science progressed, scientists decided to try something a little bit different, and they decided to see what would happen if we labeled our secondary antibodies with a fluorescent dye. Indeed, that allowed us to increase the reproducibility of the technique. Now, if we're going to go in that direction, it's probably a good idea to have those dyes fluorescing in the near-infrared range. That is because we're going to have higher sensitivity and an expanded combined linear range due to the less autofluorescence of both PVDF and nitrocellulose in the near-infrared range of light. Now, if you need more information on combined linear range, please refer to the other Lambda U courses. Now we're ready to start talking about the different reasons why scientists decide to perform Western blots so much in their labs.